Hello and welcome to section 5, the cytoskeleton. In this section, we are going to describe the cytoskeleton, compare the roles of the microfilaments, intermediate filaments, and microtubules, compare and contrast cilia and flagella, and summarize the differences among the components of prokaryotic cells, animal cells, and plant cells. So we'll start off with the cytoskeleton. We already briefly mentioned that it's going to help to maintain the cell's shape, and we can also anchor organelles in the cell. And it's really going to be made up of three different types of microtubules, I should say 3 different types of fibers, which includes microtubules. You can see some of those over here. And these are going to be the largest. We also have intermediate filaments, which will be kind of our medium size fibers. And then we have our actin filaments or microfilaments, which are going to be our smallest type of fiber. These fibers will also allow our vesicles to move within the cytoplasm and enable the cell to even move. So first, let's talk about those microfilaments a bit more. These, like we said, were the smallest, so they're the most narrow, and they're functioning more in cellular movement and provide some rigidity and shape to the cell. They can depolymerize, which means disassemble and reform very quickly, which allows the cell to change its shape and move. In our white blood cells, which are in charge of our body's ability to fight infections, this is a really important ability so that the white blood cells can move to the area of infection and go through that process of phagocytosis to eat up a pathogen. Now here we can see that this microfilament has these 2 globular proteins that are intertwined with each other and form strands, and they're serving as a track for movement of a motor protein called myosin. So if you look at this unit here, this is called a sarcomere. We find this within a muscle cell, and the actin or the microfilaments are extending out from this area called a Z-disc. And on this side as well, we have the same thing. And then these more, I guess I could call them bushy looking proteins here are our myosins. So the myosin heads will attach to the actin and will bring that actin in toward this midline called the M line. And that is how we are going to get that muscle to shorten and contract. More on that in a future chapter on the muscular system. Now with our intermediate filaments, we said this was our medium size. They're made up of several strands of fibrous proteins, and you could see that they're all wound together. This is going to be the most diverse group of our cytoskeletal elements because there are several fibrous protein types. And you're probably most familiar with keratin, in which we find in our hair, nails, and our skin's epidermis, the outer layer, and is a fibrous protein. So we don't really have a role in cell movement here. It's more about structural function. They can bear tension and anchor the nucleus and other organelles. Our microtubule is going to be our largest fiber, and we will have these hollow tubes that get created. Like the microfilaments, these microtubules can disassemble and reform very quickly. And you could see that they are made up of alpha and beta tubulin, which are 2 globular proteins, to make up the microtubule wall. In our cell, they're going to help resist compression and provide a track along which vesicles can move through the cell and pull the replicated chromosomes here lined up in the center. This is through the process of mitosis, and pull them to the opposite ends. They can also create structural elements for our cells in making up flagella and cilia and our centrioles. Now let's talk about flagella and cilia some more. Flagella are going to be those long hair-like structures, we indicated that earlier in the chapter, that are extending from the plasma membrane in order to allow the cell to move. Flagella is plural and flagellum is singular. Now our flagella is pictured here. We could see the microtubules that help create that structure. And our cilia are going to be different. They're more short hair-like structures that usually extend along the plasma membrane's entire surface. So they'll move entire cells like paramecia. They'll move substances along the cell's outer surface. For instance, in our respiratory system, we have cilia that line our cells so we can move mucus along it. And both the flagella and cilia are going to share a common structural arrangement, and this is going to be called the 9-2 array, meaning that we have a ring of 9 microtubule doublets, so you could see these kind of coupled together, surrounding a single microtubule doublet in the center.